Love to get some questions from the floor for the panelists. While you're thinking of questions, and um, uh, I'll ask the first question of the panel. So I think all three of your projects and the work that you do are completely, they're radically different from each other, and each in its own right, I think, unique. And I wanted to ask you about that, the novelty of the work that you do, and it's paradigm-changing work that you're doing, and how much of a motivation that is to you in your daily work, how important that is to you in the projects that you do. Does anybody want to take that one, Phoebe?
Yeah, I would love to, thank you. I think there's kind of two elements to this. One is that bioprospecting, looking for solutions in nature, has been happening forever. So many of our antibiotics already come from soil microbes. So in some way, we're really doing the same thing people have done before, but we're trying to change the narrative by making access and benefit sharing central to it. And so, yeah, I think we're sort of building upon what's already been there, and that's really important to not reinvent the wheel, but improve upon what's out there.
Yeah, great, thanks. Either of you like to take that question also?
Well, so from our perspective, we are always interested in looking for, again, ways that our genomic data can actually influence decisions. And our pivot to the Red Sea, which has been quite recent, is motivated, obviously, my personal interests are to discover biodiversity. And I love to characterize biodiversity. I have a long history of being a field scientist, and I'm enraptured by biodiversity. At the same point in time, we all know that this is challenging. How do we fund this? And so much of the motivation in the Red Sea Fish Program is actually motivated by looking for aquaculture solutions in a sea that is both very hot and very saline. And so how can we get fish that can survive, thrive in that, and provide a protein source for humans in a growing population in the Middle East? And so we think, using native species, right? And so this is the sort of win-win approach that is guiding our work in terms of advancing biodiversity discovery, but also finding a motivation that is admired in human progress and development.
Yeah, great, thanks. And Luca?
So what was more remarkable to me, and I think has the highest impact in conservation biology by using this technology, is that virtually breaks the divide between different fields that have to come together in order to carry on a conservation effort. In fact, in the past you had a big sequencing effort in order to characterize the genome. Then another divide was between the field work and the lab work. You usually collected samples and then you went back and have a look at what you got, and then you have an answer, but maybe it's too late because you are already out of the field. You virtually can do everything all together, or at least much easier straight in the field with this technology, and also the time required from going from I don't know anything about this species and I know even the epigenome of this species is really remarkable. And also in the time frame that takes to go from one point to the other is months, not years as in the past. So.
Yeah, great, thanks Luca. Does anybody have a question for the panel? Any of our panelists?
Hi, I have a question for Matthew. So you're sequencing birds and fish, which are the most lucky samples we can ever sequence in nanopore. Can you comment on it? Can you tell us what are your tips and tricks?
I suffer from the same issues that you do. We have blocky DNA. And with our wonderful team here from the UAE, Divin is our technical guru, we've been trying a few things. Unfortunately, I can't tell you we've had a ton of success. I can tell you a few things. Bird DNA is blocky in a different way than marine fish. It just behaves differently. They're both blocky, but they're not the same. Bird dies quickly and you can't recover the flow cell. Marine fish also dies quickly, maybe even more quickly, but with a flow cell wash, you get the flow cell back. So it's very different. What we've been trying with birds, and what I'm excited by, is heat shock and increased fragmentation. The heat shock was Divin's idea to sort of knock off, some structuring elements that might work. We seem to have some evidence that that's going to work, but we're hoping to partner more closely with Nanopore on this issue. On the other hand, we're finding that we don't need huge coverage. For bird genome assemblies, because they're not so repetitive, if we have 20x coverage, we can assemble a genome. So quite candidly, with almost every organ, yeah, I know, it's crazy, but this is this new chemistry, right? I mean, really, it makes a big difference. And we've also been improving our pipelines, and I'd be happy to talk to you about what we're doing on the bioinformatic end to wrestle as much data out of it. But in general, we actually think with a MinION flow cell, we can do most bony fish because their genomes are less than a gigabase. And for most birds with one Prometheon flow cell, and you'll note that Oxford Nanopore just lowered the price of the Prometheon flow cells. So that makes getting your favorite bird's genome even more accessible to your team.
Thank you.
Thanks. Any more questions from the floor? Okay, I have another question for the panel. So you've all talked about sequencing DNA. You're 4 times the size of NCBI reference genomes, but you're looking at methylome. And what do you think about applying nanopore sequencing beyond genomes and beyond reference genomes? New types of data and how that will impact conservation efforts? Who would like to take? You want to try, Luca?
So,
Here,
yeah, for us, the main struggle was to find the right tools, bioinformatics tools. So I would say that the 4-5 months that it took from beginning to the end, half of that time was to find the right tools that really were achieving for us the best QCs in terms of genome contiguity, mesoassembly was reduced to the minimum. So in our hands, we were quite perhaps lucky to find the right condition to get a lot of data. And despite being reptile samples, they were not that hard to sequence, even with quite high N50s with canonical ligation-based sequencing. But on the other hand, it took weeks and weeks of different tweaking with the tools to find the right pipeline to achieve comparable, if not better, quality to compared to other reference genomes.
Can I take a stab at this? So I'm going to share with you guys what I think is most exciting. So we do these reference genomes, and if you've done a reference genome and you're interested in population genomics, you're probably still using Illumina for your resequencing projects. So at the end of the day, you need population data. And right now, seems like the best way forward is Illumina, but that involves all sorts of problems when you're working around the world, when you don't have access to an Illumina sequencing center. Sending your sample, permits, it's also this sort of, we've heard several times today, this sort of helicopter approach to doing research that's sort of colonial and not what any of us want to do. It doesn't really empower local labs, doesn't work. So what I'm really excited about, and since we first started talking with Nanopore back at the PAG 30 conference in January, is how we can do low-pass imputation to remove Illumina from our data set completely. And I was really excited by Dan's talk about this topic because it's something that we in our lab have been trying to solve sort of on our own. With how do you do variant calling? How do you deal with it? The version 14 chemistry gets us to the point where it's reasonable and feasible. And if you just run your math, again, I'm going to use a bird example, I can probably do a 1x coverage genome for a bird for about $33 in consumable costs. Now that's pretty good, and that beats the pants off of anything I can do on Illumina. I don't have to send it to a center, and I don't have to wait weeks for my data. I can actually genotype in the field. And for endangered species, this is the holy grail. And so this is what I'm super excited about in this space.
Beats the pants off anything I can do with Illumina. That's my favorite sentence of the day. Phoebe, just a few seconds.
Yeah, I have a slightly
beyond genomes.
different take to Matthew. That's amazing. I'm also really excited about the other modalities that Nanopore is developing and offering. So the end goal for Basecamp is to find proteins that we can license out to the food industry or the medicine industry or whoever. And for them, it's really impactful if we're able to give more information about the protein. So being able to infer things like the expressibility, maybe if there's cofactors, what pH range or temperature gradients these things work at is really, really helpful. And so to have transcriptomic data really helps us to infer more about the protein function. And my dream is direct protein sequencing that could also read out PTMs, because that will just help us so much in understanding why won't this express when I throw it into a totally unrelated host.
Okay, great. Thank you so much. Thank you very much.